Hello everyone, I, Ravjot Kaur, biology lecturer from the School of Eminence, Model Town, Jalandhar, is hereby in front of you to deliver my lecture on Calvin cycle, C3 and C4 pathways. So let's start our topic from the process of photosynthesis. Now, photosynthesis is a biological process which is carried out by photoautolithotropes where light energy is converted into the chemical energy. So the transformation of photonic energy into chemical energy by the green parts of the plants is called as photosynthesis. Now photosynthesis has photosynthesis, the first step is the light reactions. The reactions which are dependent on the light are called the light reactions, and they are the energy producing production reactions. They convert the solar energy to the chemical energy and they utilize ATP and NADPH. That means adenosine triphosphate and nicotinamide phosphonate dehydrogenase. Now Calvin cycle, it is a light independent reactions. In this the production of sugar occurs using the chemical energy in the form of ATP and NADPH to reduce the atmospheric carbon dioxide and to synthesize glucose, that is C6H12O6. This is the formula of glucose. Now the Calvin cycle, let's start with the Calvin cycle. Now first of all, who discovered the Calvin cycle? We must know it was discovered by Calvin in the year 1955. So hence it is named as Calvin cycle. It is independent of light and hence also known as dark reaction. Now Blackman was the scientist who was first to demonstrate the biosynthetic phase of photosynthesis, which is independent of light. So it is also called as Blackman's reaction. Now the first stable compound during the process of Calvin cycle is the 3-carbon compound, that is 3-PGA, 3-phosphoglyceric acid. So another name of Calvin cycle is C3 cycle, as the carbon compound is the 3-carbon compound. During this process, carbon dioxide is reduced to carbohydrates and assimilatory paths in the form of NADPH and ATP are utilized. Now the first phase of C3 cycle is the carboxylation phase. There are 3 phases of C3 cycle. First phase is the carboxylation phase. So let's start with the carboxylation phase. It is the first reaction of C3 cycle in which carbon dioxide from the atmosphere combines with the compound ribulose 1,5-biphosphate, that is RUBP, in the presence of enzyme Rubisco to form the unstable compound, that is 2-carboxy-3-keto-ribitol, that is 1,5-biphosphate in the presence of water. The unstable compound 2-carboxy-3-keto-arbutinol biphosphate is converted to 2-phosphoglyceric acid. Now the unstable compound splits into a 3-phosphoglyceric acid. Now the second phase of C3 cycle is the glycolytic reversal. Now in this step again the assimilatory paths, that is ATP and NADPH, they are utilized. Now the glycolytic reversal includes the following steps. Now the first step of glycolytic reversal is in the presence of enzyme phosphoglycerokinase, the PGA undergoes phosphorylation and reacts with ATP and PGA to form 1,3-biphosphoglyceric acid, that is 3-PGA plus ATP in the presence of enzyme phosphoglycerokinase a compound is formed, that is 1,3-diphosphoglyceric acid, and ADP is released. Now in the second step, in the presence of enzyme triose phosphate dehydrogenase, 1,3-DPGA, that is diphosphoglyceric acid, they takes the hydrogen from the NADPH and get reduced to 3-phosphoglyceraldehyde. Now in this equilibrium with its isomer occurs, that is dihydroxyacetone phosphate. Now DPGA, that is diphosphoglyceric acid, combines with NADPH, that is assimilatory power, to form phosphoglyceraldehyde and dihydroxyacetone phosphate, which maintains the equilibrium. Now in the third step, in the presence of enzyme aldolase, now you should remember all the enzymes side by side. In the presence of enzyme aldolase, the two isomers, that is phosphoglyceraldehyde and dihydroxyacetone phosphate, they undergo the process of condensation to form 3 molecules of fructose 1,6-biphosphate. Now this occurs in the presence of enzyme aldolase. So fructose 1,6-biphosphate is formed from the isomers of phosphoglyceraldehyde and dihydroxyphosphate. Now in the next step, it occurs in the presence of enzyme phosphatase. The fructose 1,6-biphosphate lose one of the phosphate and changes to fructose 6-phosphate. That means in the presence of enzyme phosphatase, the fructose 1,6-biphosphate gets converted to fructose 6-phosphate. That means one phosphate is released. Now in the next step, in the presence of enzyme isomerase, this fructose 6-phosphate changes to the isomer glucose 6-phosphate. That is fructose 6-phosphate get converted to glucose 6-phosphate in the presence of enzyme isomerase. Now next step is in the presence of enzyme phosphatase, this glucose 6-phosphate undergoes dephosphorylation to form glucose. That is glucose 6-phosphate in the presence of enzyme phosphatase gets converted to glucose. Now the third phase of C3 cycle is the regeneration of RUBP. Now during this phase, RUBP molecules are regenerated as in the first step from the remaining fructose 6-phosphate and phosphoglyceraldehyde dihydroxyacetone phosphate molecules. Now in the presence of enzyme transketolase, so you should remember the name of the enzyme, in this case transketolase. 2 molecules of fructose 6-phosphate combines with 2 molecules of phosphoglyceraldehyde to form 1 molecule of erythrose phosphate and 1 molecule of xylulose 5-phosphate. That is fructose 6-phosphate. It's converted to two compounds, that is erythrose 4-phosphate and xylulose 5-phosphate. Now this occurs in the presence of enzyme transketolase. So fructose 6-phosphate get converted to erythrose 4-phosphate, which is 1 molecule, and another molecule is formed xylulose 5-phosphate. Now next is in the presence of enzyme aldolase, this erythrose 4-phosphate reacts with dihydroxyacetone 3-phosphate to form a 7-carbon compound. Now from the 4-carbon compound we are getting conversion to 7-carbon compound. The name of the 7-carbon compound is the pseudohaptulose 1,7 biphosphate. Now the erythrose 4-phosphate get converted to pseudohaptulose 1,7 biphosphate. Now in the presence of enzyme phosphatase, this pseudohaptulose 1,7 biphosphate, it undergoes dephosphorylation and changes to pseudohaptulose 7-phosphate. That is, one phosphate group is removed from the pseudohaptulose 1,7 biphosphate. Now this occurs in the presence of enzyme phosphatase. So 1,7 biphosphate pseudohaptulose, it gets converted to pseudohaptulose 7-phosphate. So here is the reaction: pseudohaptulose 1,7 biphosphate in the presence of enzyme phosphatase gets converted to pseudohaptulose 7-phosphate. So the pseudohaptulose 7-phosphate, which is formed from the above step in the presence of enzyme phosphatase, again in the presence of enzyme transketolase. So you should remember again the name of the enzyme is the transketolase. Pseudohaptulose 7-phosphate again react with the phosphoglyceraldehyde to form a compound, that is 5-carbon compound, ribose 5-phosphate and xylulose 5-phosphate. Now these two compounds are formed from 7-carbon compound, that is pseudohaptulose 7-phosphate in the presence of enzyme transketolase. Now next step is the both xylulose 5-phosphate, which are formed in the above steps, xylulose 5-phosphate and ribose 5-phosphate, they undergo transformation, that is they changes their isomers to form a ribulose 5-biphosphate. Now different enzymes are required for this. You should know the name of the enzyme. Now the xylulose 5-phosphate in the presence of enzyme apimerase. The name of the enzyme is apimerase to form ribulose 5-phosphate. Ribose 5-phosphate in the presence of enzyme phospholipase kinase. Again I am telling you that you should remember the name of enzyme. Phospholipase kinase. In the presence of enzyme phospholipase kinase, it forms ribulose 1,5-biphosphate plus ADP. So therefore for every carbon dioxide molecule which is entering the Calvin cycle, total number of molecules of ATP are 34. That means 34 molecules of ATP and 2 molecules of NADPH are required. So in the Calvin cycle, 34 molecules of ATP and 2 molecules of NADPH are required. Now the equation comes like this: 6 carbon dioxide plus 6 RuBP and 18 ATP, 12 NADPH. Now these 4 reactants, one is the carbon dioxide, second is the ribulose 5-phosphate, 18 ATP, 12 NADPH in the presence of different type of enzymes which we have discussed in the above steps to form glucose, that is C6H12O6, RuBP, they are 6 in number, plus 18 ADP plus 12 NADP and 10 PBI. Now diagrammatically we can represent the Calvin cycle. So the Calvin cycle occurs in the stroma of a plant cell. Now the first step is the carbon fixation. That means carbon dioxide binds with the RUBP, which is a sugar, with the help of enzyme Rubisco to form 3-PGA. Now the second step is the reduction phase, in which the 3-PGA is converted to G3P in the presence of assimilatory paths ATP and NADPH. Now next step is the carbohydrate formation, which occurs in the form of sugars, and then again the regeneration phase where the RUBP is formed. So this is the diagrammatic representation of the Calvin cycle: carbon fixation, reduction phase, carbohydrate formation, and again the regeneration phase, in which the RUBP is again made by making the use of ATP. Now next is the C4 pathway. Now this pathway was given by Hatch-Slack. So it is also known as Hatch-Slack cycle. Now what is this C4 pathway? The definition is, it is a cyclic biochemical pathway which was worked out by the scientists Hatch and Slack in the year 1966, where there is a double fixation of carbon dioxide with the first stable product, which is the 4-carbon compound, that is dicarboxylic acid. So hence it is also known as 4-carbon cycle or C4 cycle, as 4-carbon compound is formed. Now what are the characteristics of C4 pathway? Now the C4 plants which occur in the tropical and subtropical areas where the temperature is very high, aridity and salinity are common in these conditions. So the C4 cycle occurs in tropical and subtropical plants. Now the leaves of the C4 plants, they possess Kranz anatomy, in which the mesophyll cells are undifferentiated. Now there is a symplastic continuity between the mesophyll cells and the bundle sheath cells due to the presence of plasmodesmata. The carbon dioxide is initially fixed in the mesophyll cell, from where it is passed to the bundle sheath cell for the refixation in Calvin cycle. Now this Kranz anatomy, this type of Kranz anatomy doesn't occur in the C3 cycle. It occurs only and only in the C4 pathway. Now let's start with the phases of C4 pathway. Now the first step is the initial fixation. The carbon dioxide from atmosphere is first of all fixed inside the mesophyll cells. The initial acceptor of carbon dioxide is PEP, that is phosphoenol pyruvic acid. The enzyme of this carbon dioxide fixation is the PEP carboxylase. Now, PEP carboxylase is the enzyme in the initial fixation, which is the first step of C4 pathway. It produces a 4-carbon compound, that is oxaloacetic acid, hence the name C4. So, phosphoenol pyruvic acid, that is PEP, in the presence of carbon dioxide and water, again in the presence of enzyme PEP carboxylase, it forms a 4-carbon compound, that is oxaloacetic acid and H3PO4. Oxaloacetic acid is the first product of C4 plants. Now this is changed either to malic acid or to aspartic acid. Now the transport is the second step of the C4 pathway. Initially the fixation of carbon dioxide was done. Now the transport, malic acid or aspartic acid, passes into the bundle sheath cells through plasmodesmata. Oxaloacetic acid is really transported as such. Now the final fixation, that is the release of carbon dioxide. Now inside the bundle sheath cells, carbon dioxide is free from malic acid and aspartic acid through Fowling reaction. Now malic acid in the presence of assimilatory paths and to form pyruvic acid and carbon dioxide, and moreover the NADPH and hydrogen ion are released. Now carbon assimilation. Carbon dioxide in the bundle sheath is assimilated through C3 cycle. Now in the C4 plants, RUBP, which functions as final or the secondary acceptor of carbon dioxide, the enzyme involved in the carbon assimilation is the Rubisco. Now the product of this reaction is again the phosphoglyceraldehyde, which undergoes reduction into glyceraldehyde 3-phosphate, which later on gives to glucose. Now the fourth step of the C4 pathway is the regeneration of PEP, that is phosphoenol pyruvic acid. Now the pyruvic acid, which is released in the bundle sheath cells, is passed back into the mesophyll cells. Now this is changed to phosphoenol pyruvic acid with the help of ATP and inorganic phosphate. Now the enzyme involved is the phosphopyruvic dikinase. So pyruvic acid in the presence of enzyme phosphopyruvic dikinase forms phosphoenol pyruvic acid. Now diagrammatically, C4 cycle can be represented as the atmospheric carbon dioxide. It is entered into the mesophyll cells in the presence of enzyme phosphoenol pyruvate. There is a fixation and then transported to plasma in the form of plasmodesmata in the bundle sheath cells. Now there is the decarboxylation and transport again to the C3 cycle, and the regeneration of phosphoenol pyruvate occurs. Now this is the structure of the Kranz anatomy, which occurs in C4 plants, C4 pathways. Now in the Kranz anatomy, the mesophyll cells, they are not differentiated into palisade and spongy parenchyma. So this is the main identification of the Kranz anatomy, that the mesophyll cells are not differentiated into palisade and spongy parenchyma, whereas the mesophyll cells, they are compactly arranged. Now a concentric ring or a concentric layers are formed around the vascular bundles. The mesophyll cells, they are compactly arranged and they are in a concentric layers around the vascular bundles. And in addition to it, they have small chloroplasts which are specialized to perform the light reaction and C4 cycle. Now these are connected to bundle sheath cells by the plasmodesmata. The bundle sheath cells are very large in size. They have again large chloroplast which occurs in C3 cycle only. Now the bundle sheath cells are the photosynthetic cells which are arranged into a tightly packed sheath around the vein of a leaf. So what are the bundle sheath cells? They are the photosynthetic cells which are arranged into a tightly packed sheath. It forms a protective covering on the leaf vein and consists of one or more cell layers, usually parenchyma. Now the loosely arranged, the cells which are loosely arranged are the mesophyll cells. They lie between the bundle sheath and the leaf surface. So this kind of anatomy which occurs in the leaves of C4 plants, in which the bundle sheath cells are arranged in a wreath-like manner is known as Cran's anatomy. So this is the identification of the C4 plants in which bundle sheath cells are arranged in a wreath-like manner. So after studying C3 and C4 cycle, let's learn the difference between the C4 and C3 pathways. So what is the difference between the C3 cycle pathway and C4 pathway? Now the C3 cycle, the first point which you should remember always, that the primary acceptor of carbon dioxide, that means the carbon dioxide is first received by a 5-carbon compound, that is RUBP. So in the C3 cycle, the primary acceptance of carbon dioxide is done by 5-carbon compound. Whereas in the C4 cycle, the primary acceptor of carbon dioxide is a 3-carbon compound. So in the C3 cycle, carbon dioxide is accepted by 5-carbon compound, whereas in the C4 cycle, the carbon dioxide is received by a 3-carbon compound. Now the first stable compound which is formed in the C3 cycle is the 3-carbon compound, hence named as C3, that is phosphoglyceric acid. Whereas in the C4 cycle, the first stable compound is the 4-carbon compound, that is oxaloacetic acid. So this point should be remembered always, that in the C3 cycle, 3-carbon compound, and the name of the compound is phosphoglyceric acid, whereas the first stable compound in the C4 cycle is the oxaloacetic acid. Now the chloroplast in C3 cycles, they are monomorphic, whereas in the C4 cycle, chloroplasts are dimorphic. Now in the C3 cycle, the anatomy, that is Cran's anatomy, which we studied in the earlier section, was not present. But in the C4 cycle, leaves show a special type of anatomy, that is Cran's anatomy, which is present only and only in C4 plants. Now the photorespiration occurs only and only in C3 plants, whereas photorespiration occurs or rarely occurs in the C4 cycle. That means photorespiration is absent in the C4 cycle. Now for the C3 cycle, 18 ATP molecules are required, whereas in the C4 cycle it requires only 30 ATP. C3 cycle, it occurs in all type of plants, whereas C4 cycle is present in less than 1% of the angiosperms. So C3 cycle is found in all type of climate, whereas C4 cycle occurs only in the tropical condition. So basic difference between the C3 and C4 is the stable compound is the 3-carbon compound, hence named C3 cycle, whereas in the C4 cycle, the first stable compound is the oxaloacetic acid, which is a 4-carbon compound, and moreover C4 cycle are represented by Cran's anatomy, whereas Cran's anatomy is absent in the C3 cycle. So after studying the difference between the C3 and C4 pathways, now one question I will ask, by looking at a plant externally, can you tell whether a plant is C3 or C4? Why and how? The plants which are adapted to dry tropical regions, they have C4 pathways. However, the C4 plants, they do not show any characteristics in their external morphology. So therefore, we cannot say whether a plant is C3 or C4 by looking it externally. Now, which cells in the plant have rubisco? That is the bundle sheath cell. Now, name any two C4 plants, sugarcane and maize. First stable product of C4 cycle is oxaloacetic acid. Now, which compound donates hydrogen to carbohydrate in the Calvin cycle? It is NADPH. Now, expand the term RUBP. This is the question which is asked in the MCQ or in the fill in the blanks. So, you should remember RUBP. It is ribulose biphosphate. Ribulose biphosphate. Now again a question arises. So what is the basis for designating C3 and C4 pathways of photosynthesis? The number of carbon atoms in the end product is the basis for designating C3 and C4 pathways of photosynthesis. And moreover, C3 cycle is less efficient in utilizing the atmospheric carbon dioxide, whereas C4 cycle is more efficient in utilizing the atmospheric carbon dioxide. So these are the basic differences between the C3 cycle and C4 cycle and Calvin cycle. I hope that Calvin cycle C3 and C4 pathways are clear to all of you. Thank you.